Okay, today we're doing the migration protocol, day 1, and you'll need your special well plate. So remember, you open everything that's sterile inside the hood. Okay, and if you can see, these plates along the middle 2 rows have inserts inside of them. You can pick them up, and they have a little membrane at the bottom. So what we're going to do is we're going to put our cells inside the insert and put some nutrients at the bottom and see if those cells will move through that membrane. And the first thing you need to do, make sure your plate's at room temperature, and then we're going to use plain media to rehydrate the membrane. So we have 3 different types of media in this protocol. Plain media just has nothing in it, and we're going to use that to rehydrate. Then you have serum-free media, which is the plain media plus pinch strap or L-glute or whatever the media normally contains, but no serum. And then our complete media has the serum, which is the nutrients, and the pinch strap and everything, and that will go in the bottom. So first I'm going to put 100 microliters. Remember, spray the tip. Make sure you have the correct tip. And just make sure it's in the insert and not in the outside wall. And just put 100 microliters in each. Okay, and then once you've done that, you can get your cells out and count them like normal to see how many you need for the volume of wells that you're using. Okay, so grab your cells out of the incubator. And like I said, remember to note the color. These are getting pretty yellow, so almost all the nutrients are gone. It's not cloudy. So remember, I'm going to vacuum off the media. I'm going to add PBS to wash off the serum, vacuum that off. Then I'm going to add, for this one, 1 milliliter of trypsin, even if you have a large flask. And it might just take a little bit longer to get all the cells off the back. Put it in the incubator, make sure they're all off. Once they're off, instead of using complete media to bring it up to a larger volume, we're going to use our serum free. So that means there's no nutrients in with our cells. So bring them up to 10. So we'll add 9 milliliters of serum free, and then we'll count them, put them on the hemocytometer, and figure out how much of that we'll need. Because the whole point is to put those in the insert without nutrients, so they'll be drawn towards the complete media that will be in the bottom of the well. Okay, so now that you have your cell sample in the correct volume and all the inserts have rehydrated for at least 1 hour, we're going to vacuum off the media inside. So take the lid off. And I've already attached my pipette to the vacuum. So you can tilt the plate and gently touch the outside and vacuum off all the media. Okay, now I'm going to move all the inserts either up or down to get those wells free in the middle. Okay, so in here I'm going to put the complete media, 500 microliters, in the bottom of the well. And remember, RJ told us we can never put the pipette directly into here, so I need to use one of the sterile pipettes and move some media over to a tube in order to be able to put it in. Excuse me. Okay, so it's set on 500 microliters. I'm going to use the correct tip. Just going to put it in the bottom of the well. Then I am going to set it for 300. And I'm going to put 300 of my cells. Make sure you mix them if they've been sitting there a while. I'm going to put 300 in each of the inserts correspond to the correct well. And the reason we move the inserts is we don't want the pressure of the liquid in the well to come back in before we put our cells in there. So once your cells are in the insert, just gently move them over to where the complete media is. And then once you've done all your wells, cover it up and place it in the incubator.